on 10 on Saturday, and then my job, and why the prep. Yeah, I think so.
All right. Good morning, everyone. So, you guys all did a lot better on this last exam. The average was about a 78%, so about 8–10% higher than the previous ones. I did not curve it, but I did give more extra credit this time. So the grades you'll see on Blackboard are with the extra credit. I can't pass them back until everybody takes it. I still have 3 students that need to take it. And so hopefully next week I can pass them back. Any questions? All right. Today we're actually going to be switching over and talking a little bit about the microbiology of food. And so, you know, some of you guys might know, some of you guys might not be as familiar. I wasn't as familiar about all of the different aspects of microbiology that you have to integrate in making certain foods. So the microbiology of food, I guess it involves the good, the bad, and the ugly. So the good is essentially there are microbes that basically do fermentation and result in production of certain acids and products that can preserve the food. So, you know, we can produce and preserve food because of microbes. But microbes can also cause, you know, loss of the appeal, a loss of the flavor, or just maybe a really bad smell. And then there's the ugly, which microbes can result in food poisoning. There are microbes that produce toxins and things like that that can, you know, result in food poisoning. So one of the ways that microbes get their energy is through fermentation. There are different types of fermentation, and you have heard this before, but the fermentation type is classified by either the thing that's being fermented, the substrate, or the products that are formed. First thing I think of is there's anaerobic fermentation, which is fermentation by microbes that are anaerobes and they don't require oxygen. And then there's aerobic fermentation, so microbes that do require oxygen. But there are very wide different types of compounds that can be fermented, and the different fermentation products are dependent on the microbe. So one of the products of colonic fermentation in the gut is what? We literally just got tested on this. We talked about fermentation in the gut happening by microbes, and that leads to certain products. Yeah?
Is it like the
butyrate, short chain fatty acids? Yep, exactly. So if you see over here, I actually forgot my pointer in my office, but you'll see here that you have, you know, some substrate that ends up being fermented and broken down into butyrate. There's propionate right here, lactate, and then there's acetate. There's also, so these all kind of can happen depending on the type of bacteria that is breaking it down and fermenting and doing what it needs to do. So you can see here that Streptococcus makes lactate. You don't have to remember the names of the bacteria, but look at this one, Propionibacterium makes propionate. So does anyone remember what this one was involved in? Propionibacterium. Acne, yeah, exactly. So that one makes propionate. You'll see also there's ethanol. So there's some alcohols that can be produced as well. And so this usually results from yeast, where you can create ethanol. Did we talk about auto brewery syndrome a little bit? Yeah, so basically, remember when we talked about how some people that have auto brewery syndrome, they eat a lot of carbs and that gets fermented into alcohol in their gut, and then they have this hungover reaction. Some patients end up having liver failure, but that's done by the yeast. So yeasts can ferment different things, products to make alcohol. That's actually something that, if you guys remember from Bio 131, you guys did an experiment where you looked at yeast fermentation in a tube and measured the amount of bubbles produced, and that those bubbles were correlated with carbon dioxide, another byproduct. So, does that kind of sound all familiar so far? Okay, great. But we've been literally doing this for hundreds, if not thousands of years, especially with alcoholic drinks like beer and wine. And so obviously you guys are fully aware of that. But it's actually pretty interesting because even before food microbiology became a thing or a topic, lots of different ancient cultures have utilized these methods in order to, preserve their food and make food. And so, that's how we make cheeses, breads, alcohols. And so fermentation is a historically important method of preserving food. For example, the acid that's produced as fermentation reactions proceed is what makes a lot of the things that we eat not toxic. And so, for instance, acids that are produced in yogurt are really important in preserving the yogurt and not letting it go bad. Cheese and pickled vegetables actually inhibit the growth of many spoilage organisms and foodborne pathogens. So what that means is if there are organisms that are going to spoil the food, the microbes actually can produce their own antimicrobials. We call those bacteriocins. So just like we can create an antibiotic to kill off bacteria, there are bacteria that can actually produce an antimicrobial compound to help limit the numbers of other microbes that could start trying to grow and spoil the food. Those are bacteriocins. So it's like an antibiotic made by bacteria, essentially, in theory. So acid production, pickling vegetables, that's all methods of basically lactic acid fermentation. But other historically important methods to preserve food are salting, drying, adding herbs and spices, and we'll talk about that. Herbs can also produce some antimicrobials as well. And then canning or pickling, so we'll talk about those. So lactic acid bacteria and acetic acid bacteria, these basically can do fermentation and produce lactic acid or acetic acid. Lactic acid is, the lactate is the conjugate base of lactic acid, and acetate is the conjugate base of acetic acid. So lactic acid, acetic acid, think the short chain fatty acids, lactate and acetate. And so the tastes of yogurt, pickles, cheeses, and some meats are due in part to that lactic acid or acetic acid production. So taste is also influenced by fermentation. I forgot to take attendance. Why don't we do that right now? All right, Brianna, Drew, no, Bria, Henry, Anna?
Here.
Nick? Abby?
Here.
Hannah?
Here.
Rosa?
Here.
Matt?
Here.
Jillian?
Here.
Mia? Annie? Brianna? Brandon?
Here.
Sadie?
Here.
Jessica?
Here.
Jesse?
Here.
William? Marissa?
Here.
Anna? Sophia?
Here.
Aubrey?
Here.
Matthias?
Here.
Colin?
Here.
Olivia?
Here.
Braden?
Here.
Amber?
Here.
Katie?
Here.
Kennedy? Ben?
Here.
Ava? Kendall?
Here.
Emily?
Here.
Lauren?
Here.
Jennifer?
Here.
Isabel?
Here.
And Corbin. All right, sorry about that. Okay. So cheese, yogurt, kefir, other fermented milk products, these are usually products made from milk. In the cow's udder, some of you guys may or may not know this, milk is actually sterile. But while you're milking the cow and you're handling it, it rapidly becomes contaminated. And so as you're milking it, there's lactic acid bacteria that are generally on the cow's udder. And so that's obviously going to expose bacteria into the milk. And then the aesthetic features of milk change due to the production of different acids, proteases, other things that are produced. So we'll talk a little bit more about this. Kefir is a fermented drink. It's a milk drink that's similar to like a thin yogurt, and it contains live probiotics. So probiotics, if you guys remember, are microorganisms that are introduced into, your gut through food or a pill, and they are live microbes. So they are meant to just colonize that area in the gut. So the fermentation processes of these microbes that can get into the milk can change those characteristics of the milk, like the taste, the aesthetic features, all those different things, flavor. Obviously, we don't drink raw milk. Our milk is pasteurized. And milk can get contaminated from various ways, germs from the animal's feces, skin, environment, barn milking equipment. Dairy animals' udder can be infected. That's called mastitis. You don't need to remember that. The animal can have a disease. There are insects that can get in the milk. There's just conditions in the milk processing plant that can be unsanitary, cross-contamination from different people. So if you drink raw milk, will you be fine? Maybe yes, but will you get sick? Also maybe yes, so don't drink raw milk, essentially. So the natural way that milk products become fermented with these lactic acid bacteria, that's no longer used. We don't let things kind of just happen naturally. We try to take control, and we make these starter cultures where essentially we know the microorganisms that are needed. So if we know it's lactic acid bacteria, we will prepare those microorganisms, and they're deliberately used to assist the beginning of fermentation to help speed it up, produce the specific changes in the chemical composition that you need, and the properties that you want. So we select careful strains to eventually produce the desired, flavor, texture of what we want to make. Changes in the strains can change the outcome of your product, so it's important. And so starter cultures, they have to be obviously maintained carefully and protected against contamination. But essentially for commercial production of milk, you want to make a lot of it, over a short period of time. They use these starter cultures to start the fermentation process. So you guys should know the definition of a starter culture. All right, let's talk a little bit about cheese production. So cheese can be made from milk from different animals. Cheese can be classified as very hard, semi-soft, and soft. And this correlates to the amount of water. So obviously if it's softer, it's going to have more water. If cheese is coagulated, which means it's more chunky, it's changing to a solid or semi-solid state, less water. Cottage cheese is usually the easiest to make. You just basically inoculate the pasteurized milk with the starter culture, and then the culture will cause the proteins in the milk to coagulate. So coagulating means it's becoming more solid and chunky, and so coagulated proteins are called curd. Curd is essentially cottage cheese. It's really easy to make. You just put starter culture, pasteurized milk, and you let it kind of do its thing, and you get the chunk. It becomes chunky, and you get cottage cheese. The curds are then salted and pressed, and then into either blocks or reels for aging. And they eventually, to keep it chunky, they can facilitate draining of whatever liquid is left in there, and that liquid is called whey. So the whey is the liquid that's left over after milk is curdled, and that picture is illustrating what actually happens step by step. So you have, lactic acid production and renin activity will cause the milk proteins to coagulate. Lactic acid production is happening from that starter culture because they probably introduced lactic acid bacteria. Renin is an enzyme, so that's what that is, but it helps speed up the process of coagulating some of those proteins. And then those curds are heated, they're cut into small pieces, liquid whey is removed by draining, and then you can salt them and press them and age them and sell them. So making curd is really easy. It's just this first step. You add the starter culture and into the milk, and you just kind of let it do its thing. You produce the lactic acids, and it coagulates, and you make cottage cheese. But most of the other cheeses need more microbial processing, so they need to be either ripened or cured. So cottage cheese is not ripened, but for others, there's some additional processes. Different microbes that are used can help determine what type of cheese that you're left with. Remember, even a different strain can change the taste of what you're trying to make. Like I said, renin is an enzyme, basically that is added to the milk, and it's usually part of the starter culture, and it helps to hasten or speed up the protein coagulation. So does the term coagulation make sense to people? Do you guys get that? It's when things just become chunky. And then you can salt them after the whey is separated and pressed. So far we're still doing what we would do for cottage cheese, but then you can ripen them to encourage the change in texture in flavor, and ripening can take weeks to years. The longer ripening will produce more acidic cheeses that are sharper. Certain organisms produce certain characteristics. Propionibacterium shermani makes Swiss cheese. Penicillium roquefortii makes Roquefort or Gorgonzola. I've learned a lot about different cheeses by teaching this. Yeah.
When they're like hard cheeses, does that also like affect how long you would like ripen them?
Yeah, absolutely. It can definitely influence the characteristic of like how hard the cheese is. But it could be the ripening step, it could be the type of bacteria, it could be several different things. But yeah, I would think that longer ripening might influence that as well. Other questions? Okay. So let's kind of switch gears, talk a little bit about yogurt production. Basically, producing yogurt, I just had some for breakfast, is milk that's inoculated with a starter culture, so very similar. That milk is heated at really, really high temperatures. The species that tend to make milk are Streptococcus thermophilus and Lactobacillus bulgaricus, but what you guys should remember is these names of Streptococcus, Lactobacillus. These species can be produced to make yogurt. Usually that mixture of milk plus starter culture is incubated at really high temperatures. And controlling it in the incubator helps to, make sure that you're making the proper levels of the acid production in order to produce the flavors that you want. So yogurt production is similar to making cheeses, but there's this really high temperature heating step that makes it into a yogurt. Any questions so far? Okay, so I mentioned some probiotics. They're live microbes that confer a health benefit when you ingest them in sufficient quantities. So that part is actually key. If you're taking a probiotic, you have to actually ingest enough to ensure that it's going to reach the colon, where those bacteria are going to basically colonize and do microbial fermentation. So we want fermentation to happen in our gut. Remember that colonic fermentation, when we eat fibers, that is something that we can't degrade, so those fiber particles will make it to our colon, our large intestine, and then the anaerobic bacteria there are the ones that are going to process and ferment those fibers and produce short-chain fatty acids. So sometimes we can speed that up by directly putting in the microbes into our bodies and helping them reach the colon so that there's more potential for them to ferment the fibers and make short-chain fatty acids. So probiotics, there's so many different types of them. One of the things with probiotics that is still kind of under consideration is that because you ingest it orally most of the time, you would have to, the probiotics, these live microbes, have to make it through the stomach, the very acidic environment of the stomach, and then through the entire GI tract to reach the colon. Sometimes that doesn't work out. These microbes can't survive in the stomach. And so how do we ensure that those microbes are actually sticking and colonizing in the colon? So that's something to consider. You know, some of them are given in capsule form. So a lot of times that capsule dissolves while the probiotic is going through the stomach, and then the microbes die. So there are some technologies now that are considering a dual capsule technology, where you have a capsule inside of a capsule and then the probiotics are in there. And so there's some cool technological developments there, but it's still something to consider. Yeah.
Growing up, my mom always would make us take probiotics when we take antibiotics. Is there any benefits to doing that?
Absolutely, yeah, because when you're taking antibiotics, you're killing off the good and the bad bacteria. We want to keep those good ones there, and so taking probiotics helps to replenish the gut with the good bacteria while you have killed some of them off. There's a lot of good benefits to that, because you are also limiting overgrowth of yeast, you know, by making sure that there's the good bacteria being introduced and they're colonizing, and so they're taking up that space, so now the yeast can't overgrow. It also, you know, will help with recovery of your microbiome, reassembling back to what it looked like before antibiotics. So yeah, I think that's definitely something that you could benefit from. Question. Any other questions? Okay. There are different species of probiotics. If you look at, you know, probiotic, if you just grab one of these and you look at what it's made of, you'll see that the more common species are lactobacillus species, bifidobacterium species. We're not going to talk about bacillus, coagulans, or Saccharomyces boulardii. These two are the ones that I want you guys to remember. Lactobacillus and bifidobacterium are the most common species you'll find in probiotics. Does anyone have probiotics at home? All right, I want you guys to go and check what species are in there, and we'll talk about it on Monday. Bifidobacterium is also associated with breast milk, so it's a really beneficial microbe. And so the challenge with probiotics is that not all species and strains of microbes are going to have the same activity in the body. You know, so like I mentioned earlier, how do we deliver the probiotics to our gut and have them survive the process of getting through the stomach, which is very acidic and is designed to kill bacteria? One strategy is having billions of cells. So for those of you that have probiotics at home, when you go in to look at what species they are, it's also going to tell you the amount. And the amount is going to be like billions of those species in your probiotic. Another innovation is the kind of capsule, which is still an active area of optimization. And so, you know, also too much of a good thing is not necessarily always a good thing. So sometimes if you overdo it and take too many probiotics, that can lead to bloating because of the gas production. Because remember, you're making acids in these gases. So that can result in bloating and discomfort. All right. Microbes are also used to pickle vegetables. You don't have to add a starter culture here. Pickling actually originated as a way to preserve vegetables like cucumbers and cabbage, but you don't have to add anything. It uses naturally occurring lactic acid bacteria that are residing on the vegetables. So there's no starter culture involved. So the vast majority of those pickled vegetables that are sold in stores today are just simply packaged in vinegar. Vinegar is acetic acid to help pickle them. Fun fact, Dean Milner loves pickles. Kimchi. It's a traditional Korean food. Have you made it? You also Korean? Nice. Yeah, so basically it's fermented cabbage with other vegetables and seasonings. Also uses these naturally occurring lactic acid bacteria that just occur on vegetables. There's no starter culture that is used, but they have a really long shelf life, and that results in changes of the flavor. And so traditionally it's fermented in a semi-permeable vessel called an ongi. And apparently they're really cool. They're these really big like clay vessels, but they're special because they essentially can breathe. There are these tiny pores that allow the air to move through the vessel and not allow liquid, so they don't leak. But as the fermentation process is happening, carbon dioxide is being produced and it's being diffused out of the vessel, and it helps allow that reaction to continue. So as opposed to putting in a tightly sealed Tupperware that limits the amount of fermentation that can occur, it also lets fermentation go for a long period of time, and that will change the flavor. Any questions? So far, everything that we've talked about in this context of fermentation has involved bacteria. There's also fermentation that can happen by yeast, and usually that performs alcohols. They produce ethanol. Carbon dioxide is always a byproduct of fermentation. Yeasts are used to make, vinegar, bread, wine, beer, spirits. So I had asked you guys an extra credit question on the exam about which yeast or fungus I studied for my PhD. And the majority of you guys said it was Saccharomyces cerevisiae, which is incorrect. I studied Candida albicans, but for those of you that got Saccharomyces, at least you got the other yeast. The other two were bacteria. But Saccharomyces cerevisiae is that baker's yeast. You can literally buy it from the store. And it's what's used to make alcohols as well as bread. Bread actually rises because that baker's yeast is doing fermentation, and the carbon dioxide that's being produced is what allows the bread to rise. During baking, the alcohol that's produced usually evaporates because of just the high temperatures. So that's why when you are baking, the alcohol that's produced is not going to be retained. Sourdough actually has a very characteristic flavor, and that's due to the addition of lactic acid bacteria and other yeast in the place of Saccharomyces cerevisiae. So sourdough sometimes uses a starter culture to make, and so you eventually will have a culture of the yeast, but also some bacteria to help it rise. All right, any questions so far? All right, well, I have a question for you. What factors influence the growth of microbes in food? So basically, when food spoils, what are some factors that can influence that? Yeah. Temperature. Yeah. pH, environmental factors. What else? We'll go through them. So understanding these processes is really important for maintaining the quality of food and preserving them. So conditions that are naturally present in the food are called intrinsic factors, and environmental conditions like temperature and pH are extrinsic factors. And together these combine to determine which microbes grow in which foods and at what rate. So pH can be an extrinsic factor, but it could also be an intrinsic factor. It can literally be the pH of the food that you have. So another one obviously that we talked about is water availability, nutrients, any biological barriers, and if there's any antimicrobials that are being produced by the bacteria. Microbes normally multiply more rapidly when the environment is moist, there's a lot of nutrients, pH is neutral. And so it's similar to cultures of microbes that we make in the lab in nutrient liquid media or on solid agar media. Microbes like moist, nutrient-rich environments. So those are intrinsic factors as well as water availability that can influence food. And water availability is one that I want to talk about a little bit more because food vary dramatically in terms of water availability. So fresh meats and milk, they have a high water content, so that supports microbial growth. So that's why fresh meat and milk can spoil faster than dried foods like breads and nuts because they have a lower water availability. So the more water that's in that food product, the more quickly it's able to grow. Water activity is measured as this AW, activity of water. And it is used to designate the amount of water that's available in the food. So sometimes it's referred to as osmolarity, but pure water has an AW water activity of 1. And most bacteria require an AW of 0.9 or above. Fungi can handle a little bit less water and be able to grow at a water activity of 0.8. So the types of organisms that can grow and their growth rate is influenced by the water activity. So these varying availabilities of water can influence the growth rate of different microbes. So different microbes can grow as the water availability decreases. So most bacteria will need a high water activity rate in order to grow, but then there are certain ones that can still grow even after the water availability is lower. Any questions on this? pH is also really important in determining which organisms can survive and thrive on food. Many of them are inhibited by acid conditions, so a lower pH. But lactic acid bacteria are not inhibited at low pH. They're actually used, once again, in the fermentation of food. They can also cause spoilage. Remember, too much of a good thing can be bad. So many microbes are inhibited at a low pH by acidic conditions, but lactic acid bacteria, yeasts, and other fungi, they can actually survive even at relatively low pHs. So if an acidic food spoils, then it's probably being spoiled from a fungal contamination than bacteria. Because the fungi are not inhibited by the acidic environment of the food. Does that make sense? And then, like I mentioned, other intrinsic factors are nutrients, biological barriers, antimicrobials. So nutrients obviously are present in the food, and that determines the organisms that can grow. Biological barriers are essentially rinds, shells, and other outer coverings that help protect the food. So, you know, when apples or pumpkins rot, that's breaking down that covering, essentially. So that's that biological barrier. And then that causes spoilage. I mentioned that some herbs and spices actually contain antimicrobial chemicals. So those help inhibit the growth of organisms that are responsible for spoilage. So if there's those antimicrobial chemicals being produced, that's going to impact, you know, spoilage of the food. Okay, any questions? All right, we'll stop here and we'll continue with extrinsic factors on Monday.